Welcome to The Explainer. Today, we're going to unlock the secrets of life's blueprint. We're talking about how one single, pretty elegant molecule dictates, well, everything about us. I mean, from the way we look to our health, and we'll explore how we finally learned to read this incredible code. So, what is this secret code? Well, it's the molecule that writes the story of life itself. We're talking about DNA, a truly remarkable molecule that holds the entire set of instructions for building and operating, every living thing. It's why you have your parents' eyes, maybe your grandfather's smile. It's literally the story of you, just written in a chemical alphabet.
All right, let's start with the star of our show, deoxyribonucleic acid. Yeah, that's a mouthful, so we just call it DNA. This incredible molecule is the master blueprint for life. And get this, it's packed tightly into nearly every single cell in your body.
So DNA is a type of molecule called a nucleic acid. And really, it's the main carrier of all the genetic information in most living things. Now, what's wild is that scientists first found this stuff way back in the 1800s, but it took them almost 100 years to really figure out just how crucial it was, that it was the blueprint of life.
And you know that iconic shape, right? The double helix? Well, that picture wasn't clear until 1953. It was a huge breakthrough moment. You had James Watson and Francis Crick, who used some really critical X-ray data from Rosalind Franklin and Maurice Wilkins, and they pieced it all together. They figured out this elegant twisted ladder structure that, as it turns out, is the perfect way to store our genetic info.
So, how is this complex-looking ladder even built? Well, you'd be surprised. It's actually pretty simple. DNA is made of just three basic parts. You've got a sugar, a phosphate, and something called a nitrogenous base. You can just think of them as these basic building blocks that link up over and over and over again to form this incredibly long, intricate chain.
Okay, so those letters of the code, those are the four bases: adenine, guanine, cytosine, and thymine. Or, A, G, C, and T. And they follow a really simple but totally unbreakable rule. A always pairs with T, and C always pairs with G. A fun way to remember it is apples in the tree and cars in the garage. And trust me, this simple rule is absolutely crucial for everything, for how DNA stores information and how it copies itself.
But for a long time, a lot of scientists didn't think DNA was the one carrying the genetic code. They actually bet on proteins because proteins are way more complex. So how did we finally crack the case and prove it was DNA all along? Well, it turned out to be a fantastic scientific detective story that took decades to solve.
So, the first big clue dropped back in 1928. A guy named Frederick Griffith noticed that some kind of transforming principle could pass traits between bacteria. Fast forward to 1944, and you have Avery, McLeod, and McCarty showing some pretty strong evidence that this mystery substance was, in fact, DNA. But the science world needed that one final, undeniable slam dunk proof. And that came in 1952. All right, so that final piece of the puzzle, it came from this really clever experiment by Alfred Hershey and Martha Chase. What they did was they used viruses, which are basically just a little protein shell with some DNA packed inside. Then, and this is the genius part, they tagged the protein with one radioactive marker and the DNA with a different one. Then they just let these tagged viruses go and infect some bacteria.
So when they checked to see what actually got inside the bacteria, the results were crystal clear. The radioactive glow from the protein shell, it was all on the outside. But the glow from the DNA, that was all on the inside. It was game over. This was the conclusive proof everyone was looking for. DNA is the molecule that carries the instructions to build new life. Case closed. Okay, so we've confirmed DNA is the code, but what's next? I mean, a blueprint locked away in an office is pretty useless, right? You need a way to actually read it and, build the house. This is where we get to what's known as the central dogma of biology.
The central dogma is basically this dynamic three-step flow of information. First, DNA has to make perfect copies of itself. That's called replication. Next, a piece of that DNA code gets rewritten into a temporary messenger molecule called RNA. That's transcription. And finally, that RNA message is used to build proteins, which are like the workhorses that build and run the cell. That's translation. This three-step process, it's the fundamental process of life, happening in you right now.
So let's zoom in on that first step, replication. It's honestly a fascinating process. Imagine the DNA double helix getting unzipped right down the middle, just like a zipper. Each of those original halves then becomes a perfect template. Specialized enzymes swoop in and build a brand new, matching strand for each side. And why does this matter? It ensures that every single time a cell divides, the new cell gets a flawless copy of the entire genetic instruction manual.
Okay, so once we understood how DNA copies itself, scientists started thinking even bigger, like way bigger. They asked a really audacious question. Could we read the entire human genetic code from the very first letter to the very last? And this was the incredibly ambitious goal of the Human Genome Project.
3.164 billion. Let that sink in for a second. That's the staggering number of DNA base pairs, those letters we talked about, in the Human Instruction Manual. The Human Genome Project was this massive international research effort that took 13 years, all with the goal of sequencing and mapping every single one of them.
And the project was a huge success, for sure, but it also came with some massive surprises. I mean, one of the biggest was finding out that less than 2% of our genome actually codes for proteins. So what about the other 98%? Well, it used to be called junk DNA, but we now know it's anything but junk. It's actually playing these crucial roles in controlling which genes get turned on and off, kind of like a super sophisticated control panel for the whole system.
You know, the Human Genome Project also confirms something really profound about all of us. You and me and every other human on the planet, we share about 99.9% of our DNA. We're almost identical. But that tiny 0.1% difference, that's where things get really interesting, because that's what makes each of us unique. It creates a genetic fingerprint that is truly one of a kind.
And this brings us to a really powerful technique called DNA fingerprinting, which was pioneered by a scientist named Alec Jeffreys. Here's how it works. It doesn't even look at the genes we all have in common. Instead, it focuses on these specific repeating sequences in our DNA. And the exact number of times these little bits repeat, it varies like crazy from person to person. And that's what creates your unique genetic profile. So what you end up with is this unique pattern of bands, and it looks a lot like a supermarket barcode. And this genetic barcode is so specific to one person that it has totally revolutionized things like forensic science. It's been a game changer for solving crimes. And of course, it's become the gold standard for confirming family relationships, like in paternity tests.
And that brings us to a fascinating final thought. We are all, 99.9% genetically identical. Think about that. All of human diversity, everything that makes you physically you, is contained in that tiny remaining 0.1% of the code. It's a pretty powerful reminder of our shared humanity and at the same time of the incredible complexity written in the code of life. It makes you wonder, right? What other secrets are still hiding in our DNA?